Thank you for your participation in today's shelter-in-place drill. This time the drill is over. Thank you.
Good job, guys. Stay safe.
All right. So I asked you the two hypotheses this experiment is designed to test. So they're combining two things in one. So you could technically have done this in two different experiments to test the individual hypotheses separately, but they combine them into one experiment. What are those hypotheses that they're testing? Grace, what do you think? That's right. So both light and chloroplasts are required for the light reactions of photosynthesis to occur. So to do this, they're using a chemical called DPIP. And it says, what is the role of this DPIP and what molecule does it replace? So to help us to explain this, I'm going to put this picture up here. This is we talked about it in the notes, but this is a picture of the light reactions. And so this is your thylakoid and so on in the chloroplast. So for the DPIP, what is its role and what molecule does it replace? Anybody want to take that one? Carly?
It acts as the final electron acceptor for the electron transport chain in the light reactions and is replacing any DP.
That's correct. So if let's just take a second and review what happens in the light reaction. So here are your two photosystems, and this is the electron transport chain between them. Remember, at the end of the light reactions, you get ATP and NADPH made, which then go to the Calvin cycle. So when we talk about the photosystems, the light comes in, pigments absorb the light energy. Remember, these accessory pigments absorb and transfer that light energy, and all that light energy eventually goes to that chlorophyll A molecule in each one of these reaction centers. 2 electrons from that chlorophyll A molecule get excited and go to the primary electron acceptor. So now these 2 chlorophyll A molecules are missing electrons. So if you recall, these excited electrons, these guys go through the electron transport chain and replace these chlorophyll A electrons. That creates energy to pump H pluses in, which will diffuse through ATP synthase and make your ATP. So that replaces these electrons. These electrons that originally got excited here, now this chlorophyll A is missing electrons. Those are replaced by the electrons and hydrogen of water. And then these electrons end up with NADPH in the electron carrier to carry electrons to the Calvin cycle. So that's what normally happens. So in the chloroplast, you have NADP+. So in this lab, we put in here as well a chemical that's not naturally found in cells of plants called DPIP. And instead of the electrons going in this direction and NADP plus accepting them and becoming NADPH, that doesn't happen. These electrons instead go to DPIP. Why do you suppose those electrons would go to DPIP? If you have, in the chloroplast, we now have DPIP and NADP plus. What must be true about DPIP? It must be more, what have we talked about with electrons being accepted by molecules? Caitlin? That's right, it's more electronegative. For the same reason why the electrons move through the electron transport chain, each molecule is more electronegative than the next. So now when it gets here, DPIP is more electronegative than NADP+, and the electrons will go to DPIP instead of the NADP+. Now, that chemical is used specifically in this lab because when DPIP is reduced or accepts electrons, what happens to it? So if this is happening, this D in DPIP, it gets the electrons, what happens to it? Spencer? It what? It's reduced. And how would we know? Something physically happens to this. How do we know it's been reduced? Connor?
Does it change color?
It changes color. So DPIP normally is a blue color. And as it accepts electrons, it becomes more and more colorless. So it'll eventually, if it's completely reduced, it'll become colorless. And so it goes from blue to colorless. And so therefore, what we can do is measure how much light reactions are going on by looking at what's happening to the color of DPIP. Is it staying blue or is it becoming colorless? And the more colorless it is, the more light reactions are happening, and therefore we can measure how much light reactions are going on based upon that color. All right? And so, so that's what the premise of using DPIP is all about. So when we look back on the lab, so 3, how do we know, how will you know if photosynthesis is occurring? Well, what do we put there? Emily?
When you're noticing the light when the molecule is more colorless than blue, you know that more light reaction or more light transfer is taking place, so then more light reaction is occurring, and that shows that photosynthesis is happening.
Yes, absolutely. All right, and so then number 4 asked you about phosphate buffer in the water in the cuvette. So let's turn the page here before I answer that and we talk about that. Let's look at the cuvettes and just talk a little bit about that. So this, it shows you what goes in. A cuvette is like a small test tube, is all it is. All right, and it talks about here it talks about using a chloroplast solution. So let me just speak for a minute about how in the world you get a chloroplast solution. So what you would do, you take some leaves, spinach would work. You take some spinach leaves, you buy a bag of spinach or whatever at the store. You take a blender, you stuff the blender, cover the blades with spinach leaves, you pour in some water, like a water sugar solution in it, and you blend it. And it'll break up those leaves, and what that's doing is breaking up all the tissues and all the parts and so on, and actually agitating the cells and so on. And then you get this like slurry of this green spinach mix. And so then what you would do is, I would take a beaker and, I don't know if any of you are familiar with cheesecloth. It's a cloth, you can buy it in the store. It's a cloth and it acts as a filter, so you can pour the spinach solution on it, and all the big parts of the spinach get stuck in the cloth, and what drips through is the smaller part. So what would drip through is like a green watery solution, but in that green solution is chloroplasts that were once in the cells of the spinach. So it takes all the cellulose and all the bigger things out of it, the cheesecloth does, and you get this green solution with chloroplast. So that's a chloroplast solution. And so that's what they're talking about here with, and when you read here with the chloroplast. And so you have literally for this cuvette, you have these different things in them. And so let's just talk about here the phosphate buffer. So not even looking at the word phosphate, the word buffer. We talked about buffers back in chapter 3. Everybody remember what in the world the buffers did? Do you remember, John? Yeah?
It's a reaction that prevents like a radical change in concentration of anything, so it can go either way. If concentration dips too low or goes too high, it starts the reaction and returns it back to where it needs to be.
Yeah, and specifically when we say the concentration of something, buffers usually resist changes in pH. So they're resisting changes in concentration of the H plus versus OH negative ions. So this is minimizing pH changes. And so if we look here, there's a milliliter that goes into each one of these. And so that's constant in each one of these cuvettes. The distilled water, you can see here that there's 3 milliliters in all of these plus the 3 drops, and we'll talk about that in just a second. 3 milliliters of each one of in each one of these. This has 4 milliliters. But if you notice that each one of these have a milliliter of DPIP, this does not have a milliliter of DPIP. So therefore, to keep the volume the same in all the cuvettes, they added a milliliter of distilled water, all right, to keep the volume the same, and then 3 drops of unboiled chloroplast. And so there's no DPIP here, and the only one that gets boiled chloroplast is this one. Now, this one right here, this first cuvette, is what we call a blank. And so, so let's look at this here. So this is your cuvette. All right, you have your chloroplast in here. So I'm going to draw in some chloroplasts here. Now, they're microscopic, you wouldn't be able to see them here, but I want us just to see that we have that in there. And then they have the distilled water and the buffer, which are clear fluids in there. And so that would be the blank. And so every other one, these guys right here are actually part of the experiment. The only purpose, this one here, the blank, is to set the spectrophotometer. So let's talk about how we're going to tell if the DPIP goes from blue to colorless. So in the spectrophotometer, if you remember, I talked about this in our notes. Put this up here. Remember this picture? All right, the spectrophotometer, that you have a source of white light. White light is all the colors of light, all the wavelengths of light. A prism separates those into the various wavelengths. And then there's a knob on the spectrophotometer that allows you to pick which wavelength of light you're going to beam at your sample. And then it beams it at your sample that you put in and into the machine. And on the other side of your sample it records how much of that original light that's being beamed at it is being transmitted. And so remember that the more, in this case here with this chlorophyll here with pigments, the more that's transmitted, the less that is absorbed. All right, if they're inversely related there. And so that's what a spectrophotometer is. So if we go back to the lab here, we are going to use it's talking about using the spectrophotometer at a wavelength of 605. And I put next to it, what color is that? Do you guys look that up? Do you figure out what color that is? 605 nanometers. So we're not going to change that. So that's going to stay on there. So I throw up the colored picture here. Here's this picture here. 605, just into that orange there. All right. So when we look at here the action spectrum, we have some photosynthesis. Is it going on at the fastest rate? No. All right. But there's definitely photosynthesis going on in the 605 wavelength, and so that's what we're going to beam at it. And so the purpose of this blank here, the blank has no DPIP. So therefore, obviously, there's going to be no color change in a solution that doesn't have any DPIP. The purpose of this blank with no DPIP is to what we call zero out the machine. Let me ask you this. If you have this light being beamed at this sample, you have solid substances. You have little chloroplasts. Little chloroplasts floating around in here. These chloroplasts, as they're moving around in here, is that going to make a difference in the amount of light transmitted? Yeah. So if these guys are moving around here, so if it hits the chloroplast, it may not have as much light being transmitted versus if it doesn't. All right, and so on. And so, therefore, what that blank is you put it into the machine, and then you set the machine at 0% transmit. It's kind of the same idea as when we did the mass, when we had the scale and you hit the tare button. You could put the little plastic tray on there, hit the tare button, and it makes it go to 0, not counting the plastic tray, and then you can add on to the mass. In this case here, we set the blank, which is the chloroplast solution, minus the DPIP, and set it at 0% transmittance. And so then, everything that we put in and take measurements about these guys here, the only difference is the DPIP. And so any change in transmittance is a result of the change in the DPIP. Does that make sense? Okay? And so, let's look at here. Cuvette 3 says unboiled chloroplast in the dark. Before I even talk about what's in it, can somebody tell me what hypothesis you're testing there? if in that cuvette, you're gonna put unboiled chloroplast and we're gonna keep it in the dark. What hypothesis am I testing? That again? Well, it's in the dark, so this one here is testing the light, all right? To test whether or not light is present or needed to be present for photosynthesis to occur. If you look at what's in test tube 3, there's the buffer, is the 3 milliliters of distilled water, and now we have the 1 milliliter of DPIP. So you notice the difference between the blank is the same volume, but the only difference is that this one has DPIP. And so then we zeroed out the machine here, so any change in transmittance over time is due to the change in that DPIP. And so then notice here, test tube 3 is unboiled chloroplast in the light. It's set up exactly as test tube number 2. So test tube number 3 is also, these two together are testing whether or not light is present. Does that make sense? So if that's the only hypothesis we were doing, that our lab would end up with just these two, all right, as our two groups to compare to. But we're also testing a second hypothesis, and that is that chloroplasts are needed. So therefore, 4 and 5. 4 and 5 are both testing the chloroplast hypothesis. 4, look at here, it says boiled chloroplasts in the light. So everything's the same, they're getting the light, everything's the same as test tube 3, except the 3 drops are not unboiled, they're boiled. How does this play into testing whether or not chloroplasts are needed for photosynthesis? How would this help in testing that chloroplasts are needed for photosynthesis?
Because the temperature would impact the function of the chloroplast.
Okay, so the temperature would impact the function of the chloroplast. More specifically, what kinds of things in the chloroplast might it impact? And change.
The enzymes that are involved in the light reaction.
That's right, the enzymes that are involved. There are lots of enzymes that are involved in photosynthesis. What do we know about enzymes and temperature? You boil or heat them up, they denature. And so therefore, the chloroplast is still present, but you are denaturing it and changing the chloroplast, and therefore it's testing if the intact chloroplast is needed. In part of that chloroplast is all the enzymes involved. And this one is more clearly stating the chloroplast, testing the chloroplast hypothesis, because they're not putting any chloroplast in there. So no drops of chloroplast, boiled or unboiled. But notice, to keep the volume the same, they added 3 drops of water. Why? Because the 3 drops of the water is not going to affect this experiment at all, but it'll keep the volume the same. All right? So water is basically used to help. It's a reactant in photosynthesis, and it also helps to keep the volume the same. It's something that you can adjust to keep the volume. And therefore, the only thing different is either light or dark, and whether or not you're getting chloroplast or what kind of chloroplast they're getting. All right, and so, so then, what you can see here with this little setup right here, that the cuvettes will sit right here. Here's a lamp, and notice that they have this flask of water here. What is the purpose of that flask of water? Did you guys catch that? The purpose of this flask. They call it a heat sink for a reason. Huh?
It absorbs the light's infrared radiation.
That's right. All right, so it absorbs that heat energy. Our electricity, our lamps and everything electric basically is not 100% efficient. So the electricity that the lamp is using, some of that energy gets converted to the actual light, but a lot is lost as heat, increasing the entropy of the universe. All right, and so therefore this heat sink absorbs the infrared or heat energy and allows the light to still be transmitted, so these guys are still getting the light, but the heat is not gonna cook the chloroplast, all right, and so it absorbs the heat. And so what they did is then, as you saw in the table over here, is, as you read through, and they have this table here, table 4.4 here, where they did this experiment for 15 minutes, time 0, they took each one of the cuvettes and put it in the spectrophotometer, and each one of the cuvettes had the DPIP plus the chloroplast solution, and they put it in the spectrophotometer and got the percentage of light transmitted, and then they put them back into the light and let them sit for 5 minutes, and at 5 minutes they took them back out, stuck them back in the spectrophotometer, and saw how much light was transmitted and recorded the percent transmittance on the other side. And then put them back in the light and took them out at 10 minutes, then out at 15 minutes, all right? And so that was the experiment. So what I'd like you to do is underneath this, I would like you to make a hypothesis about what you think is going to happen over time for each one of these. Don't write in this because in just a few minutes I'm going to give you numbers here. But what I want you to do is look at like cuvette number 2, unboiled chloroplast in the dark. And it talked about in there how they kept it in the dark is they put aluminum foil over it, all right? And so to keep the light off of it. So what I want you to do is think about that cuvette, and underneath here, I want you to write 2 things about it. I want you to decide if you think photosynthesis is going to happen in that cuvette. And then when you decide that, I want you to then translate that to what's going to happen to the percent of light being transmitted. So as time goes on through, do not write this arrow here, I'm just illustrating here. As time goes on, is the percent of light going to increase? So more light is going to get through the sample? Is it going to stay the same? Or is there going to be even less light going through? What's going to happen to the trend over there? So you have to think about what we just talked about with DPIP and what happens to it during photosynthesis. So is photosynthesis happening? And what's going to happen to the percent of light transmitted. And then I want you to do the same for cuvette 3, 4, and 5. So you're doing this 4 times for each of those cuvettes. And you can talk amongst yourselves if you'd like, to help you to. Thank you. So here's the data to put in your data table. So let's look at this. The one in the dark. We have a slight increase in transmittance. What does that mean? So how does that relate over here to the DPIP? Over here? Charlie?
It means that, wait, sorry, what are you asking specifically?
Yeah.
Sorry.
What does the increase in transmittance here mean?
Oh, that means that the DPIP was becoming more clear, some of it was being used.
Yes. So remember that with DPIP in the presence of chloroplasts, if the light reactions is happening, the DPIP is going to accept the electrons and it goes from blue to colorless. So therefore, the more photosynthesis is going on, the more colorless the solution gets and the more light will get transmitted. So an increase in transmittance means in this lab that more light reactions are going on. Can somebody explain, do you have any idea of why, if it's unboiled in the dark, why would it get an increase here? Do you have an idea? It has to do with the setup. So in the dark, how do we put it in the dark? What did it say? In tin foil, right, and aluminum foil. And so it covers it up. But remember, every 5 minutes, what do you have to do? You have to take it out and put it in the machine. So therefore, it's really not 100% in the dark all the time. So it does usually do some photosynthesis because of that. Unboiled in the light, huge percentage increase. Is that what you would expect? Yes, because there's nothing stopping from photosynthesis. And then boiled in the light. Now notice that we get some fluctuation here with that. Did you expect for the boiled chloroplast to have much photosynthesis? No. All right. And so what I found to happen is, remember when it boils, it kind of, like I said, destroys the chloroplast a little bit, and you have more of these floaty things floating around because some of the chloroplasts are actually destroyed a little bit. And so therefore, what happens is you get this fluctuation here. Usually because these parts of the chloroplast are moving around and interfering with the transmittance of light. So depending upon if more chloroplasts one time are in front of it versus another time, you get this fluctuation. So I would say that this, even though it fluctuates, does not represent an increase in transmittance, and so I wouldn't say that there's photosynthesis going on there, and that's the explanation about the fluctuation. And then you would have expected this, right? All right, yes, okay, and so therefore that is that. All right, so we are gonna pick this up tomorrow. You do not have to do this for homework, so I want you to focus on the cellular respiration lab, okay? All right.